All right, let me start. So hello, my name is Kyle, and I'm an undergraduate research assistant at Natalie Holt's lab. We handle mostly muscle physiology research, and our presentation will be about southern alligator lizard jaw muscles. So let me first start by talking about the southern alligator lizard. The southern alligator lizard, or known as the Agaria multicarinata, is a common lizard species from the Anguidae family and is native to the North American territory. It is specifically known for its strange mating behavior, where the male lizard bites on the head of the female lizard for extended periods of time. This extended period can be up to 48 hours, exerting incredible jaw muscle endurance. Scientists are not sure why they do this. Some hypothesize that it's a form of mate guarding behavior, where the male lizard prevents the female from seeking other suitors. It can also be a form of mate selection, where the female assesses the male's jaw endurance as a criteria for mating. While the behavior is interesting, what we are more interested in is the physiology behind the jaw endurance. How does the male jaw hold the female head for such a long period of time without fatiguing? Normally, when we think of muscle fatigue, we think about how when we use our muscles intensively, they feel tired and start producing a fraction of the work we did at the beginning. For instance, when you do dumbbell curls, each curl will consist of a bicep contracting and relaxing. If you have experience of dumbbell curls, you'll find that the first few curls are always easiest comparison to the last few curls. This is because the muscle fatigues with each bicep curl, using up all the body's nutrients and building up waste, making it unable to produce enough force to support the dumbbell weight. To visually see what this looks like, we have an endurance graph that tracks the force produced of the muscle over a period of time. We can see on the blue graph that normally there are blue lines projecting in a downward trend across the graph. The bottom graph shows a more magnified version of the blue graph in which we can see that each blue line represents a wave peak. So we can interpret this using the bicep analogy again. Every time the bicep contracts, the wave peak goes up and it peaks. And every time the bicep relaxes and lets the dumbbell down, the wave peak goes down to zero, since the bicep is not producing any force when it's relaxing. Done in succession, the wave fluctuates between peaking and then zero, and each peak becomes lower because of the effects of the muscle fatigue. So that's why you get that downward trend going all the way down. In terms of the alligator lizard, the southern alligator lizard is special in that they exhibit muscle fatigue resistance that increases their endurance for biting. In a study done in 2020, it was found that the southern alligator lizard's jaw muscles contain specific contraction properties that exhibit muscle fatigue resistance. These are namely sustained force and a slow muscle specialization. If we look at a muscle endurance graph of the southern alligator lizard jaw muscle, we can see something that looks like this, which is pretty different from what the blue graph looked like. While the blue graph from the last slide fluctuates from 0 and max, in terms of the force it produces, the red graph starts kind of normal, but then doesn't reach 0 with each contraction. It forms like a wide gap under this red curve. And we call this phenomenon sustained force. Basically, what happens in sustained force is that when the red line prevents relaxations of the muscles, it will cause the muscle force to plateau. So we can see it prevents the relaxation over here, and then that force will then plateau, and then that plateau can keep going on for however long the animal wants to keep contracting. So that's how we get that insanely long muscle contraction. So why do they have this sustained force, and how is it caused? So we think that the answer lies at the molecular level with the muscles molecular properties. So when you think about in a molecular level how a muscle contracts, they contract based on its contractile proteins, actin and myosin. You can see this in this figure here, the actin and the myosin. Basically, when they interact, they cause a muscle contraction and produce the mechanical force needed to contract. And it's regulated with a protein called troponin. And this is the main protein that we're looking at here. So troponin, in conjunction with calcium, regulate muscle contraction, where when troponin binds to calcium, the contractile proteins interact and the muscle contracts. When the calcium is not bound to troponin, the proteins are prevented from interacting because of troponin, and the muscle is made to relax. This cycle of troponin unbinding and binding to calcium represents muscle contractions at a molecular level. Over here is just a figure of troponin. You can see this troponin right here and the actin right there. So an extreme example of the troponin-calcium relationship is toadfish mating calls. In a study done in 1996, the toadfish swim bladder muscles were found to be responsible for its mating call. Basically, for the mating call, the toadfish swim bladder cycles between contracting and relaxing in an extremely fast pace, where the study pinpoints that part of the reason why they're able to contract and relax so fast is because of its troponin makeup, where the troponin have a very low calcium sensitivity, meaning when the troponin binds to the calcium, it dissociates very quickly, so it makes the contraction relaxation cycle happen faster. And based on this study, we hypothesized that for our alligator lizard, our jaw muscles have a specialized troponin with high calcium sensitivity, meaning it prevents the muscle from fully relaxing and producing sustained force because of the calcium staying onto the troponin instead of the opposite happening with the toadfish bladder. To test our hypothesis, we want to quantify the alligator lizard troponin calcium sensitivity. This can be done by putting a skin fiber of a jaw muscle in a series of different calcium concentrations. By measuring force produced at each calcium concentration, we can make a PCA versus force graph, shown in the bottom right here. Basically, this graph will tell us how well troponin responds to calcium. If you notice on the right side of the graph, you see this line that signifies the toadfish swim bladder that we just talked about. And for having the line on the right side, it basically means that the troponin is less calcium sensitive. So if we want, when we collect data, we expect the line to be more on the left side. So the alligator lizard would be on the left side, while the swim bladder would be on the right side, since the alligator lizard would have a more high sensitive calcium. So the problem with we're trying to test the muscle under different calcium concentrations is the presence of sarcoplasmic reticulum. The muscle has its own storage of calcium in an organelle called the sarcoplasmic reticulum, shown in the figure right here with this big space right there. It's called sarcoplasmic reticulum. And it's basically the muscle's like own storage of calcium. And if we want to test it in different calcium concentrations to get that PCA and force graph, we have to separate the sarcoplasmic reticulum. To do this, we have to have a process called the skin. We have to make a skin single fiber, which is basically a procedure that lets us separate the sarcoplasmic reticulum and make the muscle more permeable to calcium. We basically we basically dissect the jaw muscle from a euthanized lizard, separate the single muscle fiber from the muscle, and immerse it in a variety of solutions. This basically kind of pokes holes into the muscle fiber, and it lets us put it in calcium baths and have a contraction, basically. And then to measure the force, we have this apparatus called the permeabilized fiber test apparatus. So you saw on the previous slide that there was this little machine-looking thing. Basically, that thing will be put inside this big microscope machine. And what that machine does is that it automates placing the muscle fiber into the calcium bath, which will cause a contraction. And then the machine also has a transducer and kind of like this machine that measures length change and force produced by the muscle fiber. And they also contain little wells. It's probably hard to see it, but it's right there. And it's where we store the calcium baths. And we can test multiple contractions of calcium to get that PCA force graph using these wells. And after every time we put it in the calcium bath and test it, we have to put it in a relaxing solution so that the muscle can reset back to its constant calcium concentration to prepare for the next test. And on the left, we have a image based on, we took a picture of like the image we get from the microscope. And basically we can see all like the contractions and the length changes based on the microscope image. So implications for this research. So muscle fatigue, it's not a very highly researched topic. While there's been research done in calcium sensitivity and troponin with the toadfish and other animals, they have all been mostly about fast muscle. So experimenting with much slower muscle is newer territory for muscle physiology. If we can determine the mechanism behind this extreme form of fatigue resistance we see in the lizards, we can get a step closer to understanding the nuances with muscles and maybe get some more info with the developing ways to combat fatigue-related conditions such as Addison's disease and aging. I just want to quickly acknowledge and thank the Holt Lab for giving me the equipment and the advice needed, and also the UCR mini grant for sponsoring this project. And this should be a question.